So, hello everyone, welcome to Tamali's classes. And today I'll talk to you about the genetic code. So, first I'll describe to you what it is actually. So, in my previous class, I've spoken to you about the RNA processing, remember, about the translation process, and before that, I've spoken to you about transcription. So, so I also told you about the central dogma of molecular biology, right? So we know that in translation process, basically proteins are formed. So from DNA to RNA and then RNA to proteins. So there are a set of rules which are being followed for this formation of proteins because it will. There are some rules which tells that the amino acids being formed in a protein in what sequence they are coming together and they're forming a protein. So it will start from the DNA nucleotides, the nucleotides which are present in the DNA nucleotide bases. We know there are four bases: ATCG, and in RNA again there it is AUCG. So how they are arranged and how they are forming the proteins and how they are leading to formation of amino acids and how amino acids are forming the proteins. So there are basically there are a set of rules which is known as the genetic code. In today's class, I'll talk to you about that. So watch till the end, and if you like the class, do click on the like icon and do subscribe my channel for more such videos on science topics and also on life science topics and on biology topics especially. So let's start with today's class. Also, do not forget to share it with your friends who need the classes. So let's start. So what is a genetic code? First, I'll talk about what is a genetic code. The genetic code is basically it's a set of rules. Okay. So I told you it's a set of rules that will tell us how the protein, the amino acids in the protein are being arranged. So how do we read it? This genetic code is in a form of a codon. Okay. It is made up of codons, which is also in the form of a triplet. Okay, triplets. That means there is a combination of three nucleotides which are forming a triplet, also known as the codon. So this, what are these nucleotides? They are the nucleotide bases that we have read about in DNA and in RNA. So in DNA we know ACTG. So how they are these four nucleotides, how they are arranged, three letter format, and which is known as a codon. Okay, so this each codon will specify which amino acid is needed at which position within a protein. This is done by the codon. This is specified by the codon. Okay, now this is just you can see the image out here. This image is the about all the codons. Okay, how they are formed. So I'll show it to you. So give me a second. Okay, now you can see here the first one. I told you there are three letters together. Okay, it forms a triplet format. The triplet format, the first base in the codon. See, this is a table I've shown you. In this table, you can see the first codon is U, and then here C, then A, then G. The table out here. So in the first row, you can see all the amino acids that you can see here. They are having the first letter as U. Okay, and then the second base of the codon, again you can see out here U, C, A, G, and the third base in the codon is out here U, C, A, G. So in the first row of the first, you can see, okay, let's take it one at a time. Okay, so in the first box out here you can see the first base is U, then the second base again depending on this one is U, and the in the first line U, so this is U, so U you can see. Okay, now let's talk this one about this one UGA. What is top? I will tell you later because there are some punctuations in genetic code. There are the start and stop codons. I'll tell you about it later. So just now let's focus on how a codon is being formed. Okay. So again you can see in this one you can see the first one is basis U. This U is coming from this side, the left one. Okay. And the G is coming from the top one, the second base. And the third base, the A is coming from the third the corner right hand side. Okay, this A, UGA. This is how these codons are being formed, and if you see here the names have been given of each codon, the amino acid that it's coding for, it's given. So you can see there are some codons which are there are some amino acids which are being coded by two different codons. You can see CAU is also histidine and CAC is also histidine. Glutamine, again you can see CAA and CAG. These are two different combinations, but they're forming the same uh, amino acids. Okay, that means uh, two or three codons can specify a same uh, amino acid. Okay, so we'll talk about all these things later. So again, you can see out here this AUG. This is again an important codon. Why? I will tell you later again, because it's a start codon. Okay. So similarly, we'll talk about it later. So I hope now you know how the triplets are being formed in a codon. So moving on to our next slide. Central dogma of molecular biology. I think I've explained this to you in my previous classes where I've spoken to you about the DNA, RNA. So this was proposed by Francis Crick in 1958. So it's this central dogma. This says that how genetic information is flowing from DNA to RNA and then RNA to protein. Okay, so DNA to RNA is transcription and RNA to protein is translation. Okay, so how the information in DNA is being converted to protein. So this central dogma says that. Okay, so this is expression in viruses, because in viruses there's also another step known as a reverse transcription, where the information is flowing from RNA to DNA. Okay, that's called as reverse transcription. Now, what are the properties of genetic code? Number one property is degeneracy. I've already showed you in my table uh, PPT in the slide, that how that more than one codon is coding for one amino acid. So that means most amino acids are being coded by more than one codon. Okay, so this explains the Wobble hypothesis. What is this Wobble hypothesis? We'll talk about it in the next slide. So as you have seen, I've shown you about histidine. Today I've given another example out here. The leucine amino acid is being coded by 6 different codons: UAUA, UUG, CUU, CUC, CUA, and CUG. Okay. Now, what is the importance of this degeneracy property? Now, this property provides a protection against mutations. Okay, so we know that a mutation is normally occurring in the third position of a codon. So, for example, it may not change the amino acid it's coding for. Thus, it will minimize the impact of this mutation. Since there are more than one codon specifying an amino acid, even if there is a mutation in the third base, it will not change the amino acid. Okay, so this is being protected by this degeneracy property. Now coming to universality. So this genetic code is universal across most of the organisms. You can say all organisms, but there are some minor exceptions, like in mammalian mitochondria. This UGA is normally a stop codon. Okay, but it is coding for tryptophan. Even in yeast mitochondria, this codon AUA, it is coding for, which normally codes for isoleucine, is coding for methionine in this yeast mitochondria. So there is some exceptional where the genetic code might be not be universal in some organisms. Now number three is non-overlapping. So these codons, they are read sequentially. Okay, so each base is part of only one codon. So they do not overlap among themselves. Also, they are non-ambiguous. That means each codon will specify only one amino acid. As I have shown you that more than one amino acids can specify, more than one codons can specify one amino acid, but each codon will specify only one amino acid. That means if AUG is methionine, AUG will not be any other amino acid. Okay. Now, next is punctuation in genetic code. As I told you that there are some punctuations, like there are some start codon and some stop codons. So start codons are those codons, which will signal the beginning of translation. That means when the translation step is occurring, so I've shown you in my, I've explained to you in my previous class in this translation process, that when this tRNA is bringing the amino acids, so how will it identify? How will it start to produce those amino acids? How in this mRNA? So first, the start codon is being identified. Start codon means that codon which will signal the beginning of translation. As soon as that meets as soon as when they are coming together, tRNA and mRNA, the amino acids, when they're coming together, as soon as it identifies the start codon, it will start the translation process. So basically, AUG is a start codon and it codes for methionine. Okay, then we'll talk about the stop codons. So there are some codons which are used to stop the translation process. That means when these codons are identified in the messenger RNA, immediately the translation stops. That means it will not code for any amino acids. So there are three: UAA, UAG, and UGA. So these three are the stop codons. Okay, you can see below there's a diagram out here. There's a table out here. You can see. So picture you can say. So AUG is the first codon. CAU is a second codon, GAC third codon, UCG fourth codon, and UGA fifth codon, which is a stop codon. Okay, so as soon as the stop codon is being met, it stops. Okay, now coming to triplet code, I've already explained this to you in my earlier slides. I'll just give you a basic again. This genetic code is a triplet code. That means there are three nucleotide bases coming together, which will specify some amino acids in DNA and RNA. Also, as I showed you the table of genetic code, it is consisting of 64 codons. Okay, that means all the possible combinations of these four nucleotides A, C, G, U. Because why not T out here? Because DNA is being converted to RNA, and from RNA we are converting it to proteins. And since this is we're talking about the protein combinations, the amino acid combinations. So out here we're talking about ACGU, not ACTG. Okay, so ACGU there are 64. There are codons total, and there are these are they form the maximum, like the combinations are being formed. Okay, so 64, the genetic code is made of 64 codons, which are the possible combinations of this four nucleotide bases. Okay, so now I told you about stop and stop signal and the beginning, the start signal I've told you. So what does it do? That also I've told you. So sometimes mutations can occur in genetic code, which can cause an early stop codon to come, and this might have an impact in the translation process and also on the transcription process. Okay, now you can see the picture out here. You can see if there's an incorrect amino acid, is if it's being incorporated, then it may lead to a malfunctioning of protein. Okay, it can lead to a protein malfunction, you can say that. So out here you can see this histidine is in place of histidine, it's coming to proline out here. So again, it can lead to a malfunctioning, it can lead to a different protein. Okay, now we'll talk about Wobble hypothesis. It's very, very important, and questions come from this part in your CSIR exams. Okay, so this Wobble hypothesis is very important because it explains the degeneracy of the genetic code. Okay, now see, this genetic codes, they are degenerate, as I've told you that many amino acids, like many codons can code for one amino acid. Okay, so this was given by Francis Crick in 1966, and what does it state? It states that the third base in a codon, we have seen that it's a triplet. The third base in a codon can wobble. Okay, wobble means it will it will turn. Okay, it will turn and it will not form the usual Watson-Crick base pairing. So we know the Watson-Crick base pairing, how it is forming a helical structure and all. We have seen that you can check the classes of my DNA and RNA where I have explained the structure of DNA. I've shown you how Watson and Crick base pairing is being done. So in Watson and Crick base pairing, whatever the format is, the formation is, that will not be followed because of this Wobble hypothesis. So it will form a non-Watson and Crick base pairing. Okay, with the first base in the anticodon tRNA. So Francis Crick said that the 5' base on the anticodon, which binds to the third base in the mRNA, was not as spatially confined as the other two bases, and thus could have non-standard base pairing. So this is the postulate that was given by Francis Crick. Okay, so this explains that why multiple codons for the same amino acid often differ only at the third base. We have seen in the table, right, that the third bases are changing. Okay, see, I'll show you again out here. See out here, we have seen in glutamine CAA, CAG. CA is same for both cases out here, right? See, I'll show you I'll show you here. See in the table out here, you can see anyone see glutamine in example, like it's CA is same from the first two, but A is different, G is different. Similarly, you can see in lysine again, the third base, A is different, G is different. You can see in RG9 again, A different, G different. You can see all this out here. The last third base is different out here. Okay, so this is what? This is degeneracy, right? So this is again because of this Wobble hypothesis. Okay, so this explains that why multiple codons for the same amino acid, why they differ at the third base, because they're not following the Watson and Crick base pairing. Okay, so this Wobble hypothesis will explain the degeneracy of the genetic code. Okay, now importance of genetic code. Why it is important? Because see, we know that proteins are very, very important and necessary for life. So therefore, Our cells need this genetic code. It needs those set of rules to form the correct proteins that are important for our life. Also, we need genetic code to study the genetics. Okay, even for genetic engineering, to understand how this genetic code works, we need to learn it, and then once we know how it works, scientists can manipulate this genetic code and it can form the desired traits in an organism. Suppose we need this certain trait, that means we need a certain type of protein. So when we know that this amino acid is required for this protein, we can manipulate the genetic code and we can produce what we require, the characteristics we require in that organism. So this has the potential to revolutionize medicine, agriculture, and all other fields. Okay. Also, for evolution studies, how evolution is occurring, what is changing, because we know when evolution occurs, when changes occur, there is some protein changes, right? So we can study that. Also, we can compare genetic codes of different organisms and we can see how closely they are related and how they have evolved over time. Okay, so at the end, I've okay, one more thing I've missed out here. How genetic code is read. The genetic code is read from the 5' direction to 3' direction in mRNA. So remember that. Okay. Now, here I've given some questions, MCQ based questions for you to solve. Do solve these questions, and if you have any doubt, do comment below, and also let me know what the answers are. And you can solve and you can know how much you are clarified in this topic. And if you like it, do not forget to subscribe to my channel and keep watching the videos which you require for your exams and also for your knowledge. So thank you for watching the video. See you again in my next class. Thank you so much.